Hi, everyone. Welcome to the ISME Early Career Scientist Committee. I'm Inzhao Wang, one of the committee members. It is really a pleasure to have the ISME Ambassador of China, Professor Fengping Wang, to a series of video conversation called the ISME Conversation. The goal of the ISME Conversation is to having video interviews with senior researchers and to get to know their research and their experiences during their early career time, as well as their scientific life. So it is a great pleasure to have Professor Fengping here today. And I would like to have a brief introduction. So actually, I knew Fengping when I was a PhD student at the AGU meeting, and then worked with her during my postdoc time. And I really learned a lot from her. And I'm sure that you also will this time. So Fengping graduated from Central China Normal University and got her PhD at Huazhong Agricultural University, then went to Osnabrück University for postdoc at Germany. After that, she got a position at Third Institute of Oceanography in 2003, and then moved to Shanghai Jiao Tong University in 2009. She is now the ISME Ambassador of China and the committee member of International Ocean Discovery Program, Deep Carbon Observatory Deep Life Committee, and also the executive director of International Center for Deep Life Investigation. And she won a lot of prizes in microbiology research field. So thank you for coming.
Yeah, thank you, Inja, for inviting me to have this live conversation. I think this is really a very good chance to know more people, to know more research, and I hesitate to say that I have many experience, but I would love to have the chance to chat with you, Inja.
Thank you. So, okay, I would like to go to my first questions. Okay, so what is your research about? How did you decide on this topic?
I think as you briefly introduced in my CV, I'm mostly working on deep sea and subsurface microbes. And it's fascinating to know who are living there and what they are doing and with what kind of mechanism. And eventually, I also wish to know the contribution of these microbes to the Earth's elemental cycling. How I decide on this topic? Actually, this is a long story.
Wow.
It took me a long way to my current research. I did my PhD at Huazhong Agricultural University, and there I mostly study rice genetics. And so there's nothing or very little relation with ocean science. But after I got my PhD, there is a chance to go to the Third Institute of Oceanography to do some research. There's a position there available. So I think, wow, why not just try? So I took that opportunity to go to the Third Institute of Oceanography. And meantime, I got a postdoc offer from Germany to do some soil microbiology research, actually mostly on biochemical research on streptomyces. Yeah, so I think microbes are very important, not only on land, but also in the ocean. So I think probably that's a very good chance to know more about microbes. And then I get more experience. Then I can move and to do more research on microbial in the ocean. So you say that that's really took me a long time and a long way to my current research.
Yeah, but you have seen different views during your history of your research. you experienced a lot of different things that probably will open a lot of doors in your research.
Yeah, that's also the part I love it. That
Yeah.
there are always new things. Yeah,
Yeah, that's right. So
yeah.
my next question is, how did you decide it was a time to transition from the postdoc and start your own group?
Um, I cannot say I know it exactly. I decided it was the right time to do the transition. Um, it seems to me not a decision by me, but a very natural transition. And it's something like there's a chance. And then especially when I started, it was the chance that in China, it launches projects to do deep sea research. And then the chance is there. So why not? Just go
Yeah,
to try.
yeah,
So to me, it's not really a very big struggling decision to be or not to be. You know, it's more like very natural that here is the chance, and then you can get it, and it's very interesting, and so yeah, so you just do it. So for
yeah,
me, in my case, it's very natural. Yeah, I,
yeah.
I don't know. Yeah, that's helpful for the others, but it's the case for me. Yeah.
Yeah. But you know, when I work with you, I can feel your passions and your science, the dream for science, and also that you know, like I the achievements that you have made. So I was wondering, what was your strategy to build your team when you started, and how many students and postdoc did you recruit?
Oh,
That's a
that's
long time.
that's a long time. Let me think. I think when I started, I just had one master student, and you I would say it's still a very difficult time for research in China at that time. Of course, you get more chances. For example, for me, it's very natural that you can move from postdoc to a faculty position, but the condition is really poor. Yeah, the equipments are poor, and including yourself, maybe you are not really ready, and you don't have many students to start with.
Mm,
The only thing I think I remember I need to work very, very hard.
yeah.
Yeah, because I only have one master student.
Wow.
And so I need to do experiment together with her
Yeah.
and design every procedure, protocol, everything. Yeah, it's really very, very tough. um yeah, including also you need to improve yourself, and I mean it's really very challenging to me is that actually I also you know my background, I'm not in ocean microbiology.
Mm,
um, I mean I was trained in rice genetics. So either there's no background training of microbiology and also no training on oceanography. Yeah. So I need to learn all of this together with the students. And I think this is also the part I love it, that my job, that you got a lot of new things challenging, but also you found yourself really improved, and you have new discoveries and have new friends. Yeah, and enjoy a lot.
yeah.
So
Yeah.
I think that's I enjoy a lot. Although I worked very hard, I I have to say I worked much harder than I am now. Yeah,
Yeah.
yeah,
Yeah, I can feel that it's extremely difficult at that time. I think it's things are getting better today because they have a bunch of money and they can also get the postdoc and also students. I think but I think at your time the spirit is very strong and you have a good friendship with the students.
yeah. I would say that right now, there are so many help. Also, I would like to say that during the past, you are not alone. You
Mm,
have so many support from other, including that time senior scientists,
yeah,
and also support from friends.
yeah.
Especially, I would like to mention that at that time I get many support from our um, international mentors,
Oh.
friends. Like I would say that from Japan,
Mm,
Chucky Kato from
oh.
James Tech at that time, he came to us to get the training course, and also that Dognat Batnet, and she also come to have the workshop, and we really learn a lot from them. And I would say that have friends and have support is really important.
Yeah, that's right. That's a lot of people behind you. So I think my next question is probably you have answered. I'm not sure because the next question is what was your biggest challenge at during your early career time? Is it the I mean the not too much students or postdoc?
I would say it's simply no enough time.
Yeah.
Yeah, you need to learn so much. You you still not mature, and there's so many new things you need to learn. And on the same time, you have family,
Yeah.
and I have a boy, a
Hmm.
son, and even I have to say that during my very early time, I even don't dare to have a kid.
Oh,
It's
wow.
really
That's challenging.
yeah. So yeah, I really don't feel I have time.
Hmm.
So yeah, that's really I think the biggest challenge that you need to really balance your time and also be very focusing. Yeah.
That's right. And so looking at back your history and when you look about earlier time, so what is the most important achievement at that time, you think?
Uh, I think to get started is that um, I got a big fan from camera
Oh,
that is yeah um, inst- like a center or institute for ocean mineral and science research association. And so after you get funded, and that means you are secured to do deep sea microbiology research. Yeah.
yeah. Yeah,
So I
that's
think
right.
that part is very important to me. And also important is that I went friendship with a lot of colleagues,
Hmm.
um, that including, I would say that, for example, from Huaiyang Zhou,
Oh,
right
yeah.
now he's at Tongji University,
Yeah.
and Chuanlun Zhang.
Hmm.
At that time, he was in the States, and right now he's at China at Shenzhen, uh, at a university at Shenzhen, and Jill at Oklahoma University doing geochip. Uh, and also, yeah, my husband, Shao Xiang, yeah, together. Yeah. So I think after we got the funding and got a lot of very, I mean, the support from colleagues, and then we decide to do something really cool that we used the geochip developed by Joe in his lab to monitor the dynamic change of microbial community from a newborn proto-chimney to the mature chimney. That is really collaborating work that at that time we don't have ROV or HOV down to the deep ocean. So at that time, Huayang Zhou got us collaboration with Hui and used Arvin to dive to the Wandafuka Ridge. And at there, we do the experiments that they move the chimney, and at the removed chimney, they put like a cap on the top of a venting field.
Cool.
And then after a few days, and then a newborn chimney was obtained. And then yeah, so they got the proto chimney, and then they also got other chimneys like material and dead chimneys, and then we can check using including geochip and other advanced technology, including like mineral research, and also that quantitative PCR and those methods together. And then we can investigate the change of the community from just the newborn, the proto chimney, to the have matured and also not, active chimney through this process, and how the microbial community change with the mineral composition. I think that part, I love it a lot, this work. It's not only that you need really different high technology, and also that it's really interdisciplinary. Yes. And I really think through this work, I get experience that geology people, biology people, and also like the other field people can work together to understand one thing. I think that part, that work really give me a lot of, very important experience later when I designed other research. I'm always think that when we want to understand microbial roles, we cannot only focusing on microbes itself. We need to integrate other fields together. So I think for this work is really a true starting of during my early period of time.
Wow, that's a great experiences. And I also know that you have a dive into the deep seas. Could you share that experiences with us? Because I think that's really exciting and scary.
I would describe my first dive with Arvin, but that's not very early. It's already like in the year, let me think, 2009 or 10. I cannot remember it exactly right now. That dive and that cruise I also love a lot. That is a dive a cruise with chief scientist is Anna Louise Reisenbach. Yeah, and she got a fund to do research on Guaymas Basin, and she invited me to join her in the cruise. And then during that cruise, and I obtained a prestigious chance
Wow.
to dive. Yeah, yeah. I still remember that. Yeah, I dived to more than 2,000 meters.
Wow.
Yeah, and I still remember. Yeah, the first thing I saw in the deep ocean at first, it's dark, and after it bottomed at the sea floor, and of course there are night, yeah, from the RV, and there's suddenly something come out,
Wow.
and really, yeah, really, wow. Yeah, and that's really my first time see the life in hydrothermal vents, and they are so beautiful. And also it amazed me that they can, especially the American scientists, could do so precise research, precise sampling. You know, at that time in China, to do deep sea research is very, very difficult. We don't have any of this high tech. The only samples at that time I can get is just from like a TV grabber, whatever you grabbed, and then you do whatever, yeah, that grabber got to you.
Yeah.
So it's very hard, very hard. And then at that time, I already so amazed, and also like asking that when could I get a chance, or China get this high tech ROV, HUV, yeah, and then we can do the core science. Yeah, yeah, that's really one thing. I think a very big change. Yeah.
Yeah, that's very exciting. Can you actually list two uh, two papers that you most like up to now?
Yeah. The papers I like most. You mean in my early
Uh,
career,
the
or
entire
my full time
career.
entire?
Yeah.
Wow, that's a lot.
Yeah.
Kidding. Yeah, I think the PNS paper I love because it's a starting of my interdisciplinary research, and I really later got experience to design yeah, those kind of research. And the other papers I think is about Basia acuta.
Yeah, yeah.
Yeah. Yes.
Yeah.
Yeah. I would name the paper at ISME 2014, that paper. That paper, um, you know that Basia acuta formerly described as MCG
Yeah.
and Mycenius crown acuta group.
Yeah.
And when I started my research, I told you that we cannot get precise samples with precise physical chemical characters. We often just by TV grabber grab some sediments. And then I got a chance to analyze a lot of different sediment samples to check what microbes are there. And then I found that MCG was one of the mostly abundantly distributed microbes in marine sediments. In almost every samples I got, I detected MCG.
Wow.
And then, yeah, but at that time, we we don't know what they are doing. And then it took a long time, at least 5 years, to find to get more genetic materials from this MCG, and we constructed libraries like Cosmate or those libraries. The libraries, you know that you have a vector, and using the vector you got the DNA from different microbes. And then if you determine one clone got the MCG16S, and then you sequence those cosmate or those plasmids or vector, and then you get to know more about these microbes. So we got a lot of time. We really spend a lot of time to construct a library for marine sediments. After that, we did that, what is it called? That hybridization with the 16S or PCR to check which colony got the 16S. And then after we got that clone, and then we sent it to company for sequencing. And from this work, we got two very interesting findings. One is that we found one clone have the 16S of MCG, and also it get a gene for photosynthesis essential gene that bacteria chlorophyll A synthesis. That gene has never been found in archaea before. So that's the first time found an archaea have the gene BCHG. And then we expressed this gene and then get the function, and it's really functional that is really a functional gene. It's not only a putative gene for that, but it's very tricky to really think about that, the function of BCHG in Bacillus acutata in MCG, because it's in the dark marine sediment. So why it needs this gene? Maybe in the cell it have other function. This is very interesting parts, and I'm still, like, even in today, I'm still wondering if archaea really play some roles in the process of photosynthesis.
That's right. I know that.
Even for today, I'm still checking in other archaea, and if we can find more genes in the process of photosynthesis. And that paper was published on ISME, and that was my first ISME paper.
Wow, I know that's a famous paper.
That's really one of my favorite ISME paper, and I'm still intriguing to find what's the role of this BCHG really in archaea, and also if there are more genes in photosynthesis.
That would be cool. Probably an altered version of original of photosynthesis.
We don't know, maybe. And then later in the actually there's a mix in the 2014 ISME paper from us is about we got more clones, and then through a more detailed or more robust phylogenetic analysis, we point out that MCG actually not belong to Crown acute. It should be another phylum.
Yeah.
So yeah, we name it putative layer spatia acuta. So right now there's many papers, yeah, and research also including us on this like widely distributed but functionally undetermined archaea group. Yes.
Yeah. I like that name. I think that name is a kind of a mysterious.
Oh, Basi.
Yeah. And so, what do you think is the most important characters that leading you to your achievement today?
Oh, character.
Or aspect?
I would say probably first is persistence.
Yeah, that's right. Yeah.
Yeah. And the other part maybe I'm optimistic.
Yeah, that's right. I can feel that. I like that.
Yeah.
Yeah,
So yeah.
that's very charming characteristic. I can totally feel that.
I mean and this is also I feel that I can share with people that no matter what you will do or are doing, certainly there will be difficulties, but if you choose, I mean, do what you like or you love to do, you can, I think that is kind of persistence. You can be persistent and also be optimistic that yeah, you can you why not think in another way that it's very lucky that you can do the work you like or you love. That itself, it's a very lucky thing.
Yeah,
Yeah.
that's right. Yeah.
Yeah.
So, about running a lab. So, what did you learn later that you wish you know before? I mean, when you start your group.
Mm, let me think. I think probably if you could set a few goals for your lab development,
Yeah.
that would be good. Yeah, because when you start running your lab and there suddenly the transition from a student or postdoc to a staff, and suddenly there'll be many disturbing or distracting things. Yeah.
Yeah.
Um, so it would be very good if you can put a few goals and then check like each year or 2 to 3 years to say if you can get to your goals.
Yeah.
Yeah, and then I feel that uh, can make you always on track and check yourself if you are on track.
Yeah.
Yeah. So I think this I would say if I run my lab from the start, I would set a few goals. And also, if I determined the goals, and now I will be more focusing. And you know that there will be many chances or opportunities, and that can also be distracting sense.
Yeah, that's
Uh,
right.
yeah, that can lag you away from your major goal. So if you can set a goal and then you can be more focusing, and then you can just give up a few other things that are not so important to your major goals.
Yeah,
Yeah.
that's very important. Yeah. And so, if you could change one thing about the science or academic, what it would be?
Yeah, I actually, what would you mean that change one thing about change my my career or what?
Oh no. So this is about the science committee or the sounds like a regulation or something, or the uh, atmosphere, anything that you can think, or the technique, or the
Mm-hmm. Um, um, Let me think. Um, I think probably like the way we do deep ocean science today, and especially when we think about we measure the rates or the activities of microbes, and then or the roles of archaea in the ocean, we still need to do it on the deck or go back to the lab. And so right now, we with our colleagues together, we wish that in the future we can do more things, just under the ocean. Yeah,
yeah.
do you mean this kind of things or about a
Oh.
science committee?
Anything that just to come to your mind. I think that you mentioned the question that is very hard and very frontiers in the microbiome ecologies that we see the microbes in nature directly.
Yeah, yeah,
Yeah, that's very
and
important.
to develop different techniques to
Mm.
detect. For example, one thing I'm very much now focusing or interested in is that to check the different gene expression in one single cell. Yeah,
Wow.
so right
Mm.
now, yeah, we check, we when we see the cells activity, actually, we not see one cell, we see a population, and
Mm.
within the population, different cells have different activity actually,
Yeah.
and they behave differently, and we are somehow lacking of ways to differentiate or to target a single cell with different gene expression. So I think this way probably we can do something.
Yeah, that's very challenging and charming.
Yeah, and I would like to say another thing in the science community is that I hope I can do something in the science community in China, especially in the microbial ecology society, nature. I wish to encourage more female scientists, yeah, in the field
Yeah.
and to take the lead. Yeah,
Yeah. That's very important.
yeah, probably I can do something, and also I'm collaborate with some other women scientists
Mm.
together to host a series different workshops and and symposium, and yeah, to to have female scientists to voice out. Yeah.
Yeah, that would be great. Yeah. So, so what is the most important advice that you gave to your postdoc when they leave your lab and start their own group?
I think um yeah, I really don't think I can give very specific advice. Each person are different and each generation are different.
Yeah.
But one thing I would say, um, looking at myself, my parts, I always feel that I got so many helps from others.
Yeah.
So I think one thing is that be generous to others and
Mm.
then make more friends.
Yeah, that's right. Friends is good. Yeah. So we can away from the science. So except the science, is there any other things that you like most?
I like most.
For example, some kind of music or any sports.
I think I like music when I was young, listen to a lot of different music. But right now I feel I'm getting more and more lazy. I
Probably just because you're busy.
no, to music sometimes I will go to the concert to hear the symphony.
Wow.
And sometimes, and also I hear pop music.
Hmm.
Yeah. So, but mostly, I think going to sport, I just like jogging.
Hmm.
Yeah.
Yeah.
Not interesting spare time life.
So probably this is the last of my question is how would you relax yourself during your work?
Um,
I mean, it's probably a very tough question for a chief scientist.
no, no. I mean, I mean, um, everyone needs to balance from between work and I mean daily life, relaxing.
Yeah.
And for me, I can just do nothing,
Hmm.
just totally be empty,
Empathy.
think nothing, do nothing.
Yeah.
Yeah, maybe just walk around and sometimes have tea with friends.
Yeah.
Yeah, and chat.
Wow.
Yeah,
That would be very great.
yeah,
Especially you have so much friends.
yeah. I have many friends, and then yeah. So that means you also need time to keep the friendship. Yeah,
Yeah.
yeah,
And during that time, probably a great scientific idea will come out.
yeah, yeah. Sometimes, yeah, it's just pump out. Yeah,
Yeah. Yeah, thank you. Thank you very much for this interview. And think we can learn a lot from this interview. And thank you so much personally that I still work with you. So it's my privilege. Thank you.
thank you, Inja. It's really nice from you. And also, I really feel that I also got a lot of encouragement from you and other
Thank you.
like postdocs and students. And I think this is also one of the big support. Yeah, yeah.
Yeah.
I would love to continue to with all the colleagues, friends, and also the students to find novel things and
Yeah.
to enjoy the life and to enjoy the science world.
Yeah,
Yeah.
that's right. Thank you. Thank you so much for your
Thank
time.
you. Yeah. Okay.
Okay, bye.
Bye.